Welcome to Executive Insights for Life Sciences Innovators, and thanks for joining us. On this podcast, we'll explore leadership, innovation, and growth in the life sciences sector. I'm Don Alexander, founder of GeneCoda, a boutique executive search firm dedicated to connecting visionary leaders with life sciences companies shaping the future of therapeutics and scientific tools. On each episode, we'll dive into candid conversations with industry pioneers, uncovering strategies, insights, and real world experiences that drive success in our industry. If you're interested in presenting as a guest of a future episode, please scan the barcode image at the top of my video frame and follow the instructions. Now I'm pleased to be joined by Ben Scruggs, the CEO of Altis Biosystems. Welcome, Ben.
Thanks, Don. It's a pleasure to be here. Appreciate you making the time to chat.
Absolutely. Thanks for your time. Ben is a partner at Hatteras Venture Partners, where he focuses on biotechnology and life sciences tools companies. He's been involved in building and scaling transformative healthcare companies across a swath of therapeutic areas, including inflammation, oncology, neurodegeneration, ophthalmology, cell therapy, and gene therapy. Ben currently serves as the CEO and chairman of Altis Biosystems, a human stem cell derived platform company advancing more predictive in vitro models for drug development. Prior to Altis, he led strategic initiatives at Tune Therapeutics, a biotechnology company pioneering epigenetic editing. He has extensive experience working alongside scientific founders and academic innovators, and also serves as manager of L2 Ventures, a seed stage fund supporting startups affiliated with UNC. Ben currently serves on the boards of Invention Therapeutics and Verilux Therapeutics, and is also a board observer at GeneCentric Therapeutics and Create Medicines. He's previously served as a board observer at Dropworks, acquired by Biorad, OPEX Biopharma, acquired by Shionogi, and Rodin Therapeutics, acquired by Alkermes. Before joining Hatteras in 2016, Ben completed a postdoctoral fellowship in the Epigenetics and Stem Cell Biology Laboratory at the NIEHS, where his research focused on transcriptional regulation and stem cell biology. Ben received his PhD in molecular biology, cell biology from Washington University in St. Louis, supported by an NIH NHLBI fellowship, and he holds a BE in biomedical engineering from Vanderbilt University. So that's quite an impressive resume, Ben. Um,
Thanks, Don.
Absolutely. So let's dive a bit into your career journey and leadership philosophy. I guess starting back maybe a few years, what first drew you into life sciences and translational research?
Yeah, life sciences, translational research in particular. I mean, for me, I actually have a really strong affinity to the bench. I love bench work, and was fortunate to do my PhD at WashU in St. Louis, but I think that had a really strong impact on me and working at a center that not only does great research, but is also a major medical center. And so working elbow to elbow with people who were treating patients on a daily basis really made me start thinking about the ways that basic research can be translated out of the lab and actually start helping patients. And for me, part of that impact and light bulb really went off thinking about some of the newer modalities that have popped up in the past decade plus. I was doing research, basic research in the field of RNA before that became as commonplace as it is today, seeing some of the early companies that popped up in that space, companies like Alnylam, and starting to see how things like what I was doing at a basic level could really be translated into therapeutics. And from that standpoint, that was really a motivating factor of trying to think about, while I have a love of what happens at the bench, how can you take those things and take novel innovation and turn that into any type of new product that can be used in a patient care setting. And that's kind of been the springboard for me and really the common thread of everyone that I've worked with, and why I love working with founders and want to work with companies directly in terms of being able to take novel technologies and help turn them into new products for patients.
That's fantastic. Really great overview, Ben. Thank you. So, when did you realize the industry needed a better intestinal model?
um, no, I think that the aspect for me was thinking about the some of the gaps in preclinical drug development in terms of efficiencies of being able to take a novel therapeutic and the amount of time that it takes to get something into the clinic. And for good reason. The preclinical development is a rigorous process, but I think the number of companies that I've worked with at the early stage and really seeing that there can be disparities between different types of species and all of the different animal models that are used, be that differences between rat and dog or with non-human primates, that having new systems that are human-based is really a motivating factor for me to help accelerate some of the new types of tools that can be used in either in vitro models or more broadly speaking in new approach methodologies, so that there can be a quicker time to answer of should a new compound continue to be developed or can you triage those compounds? I think that has been a motivating factor for me in working with Altis and developing, commercializing a model system of the gut. And I think the passion for me, we think of Altis as a company that uses stem cell based technologies, starting with human based material, so that you get that prediction of how human biology should actually happen in the preclinical setting before you actually have to take something into patients. But the need for a gut based model really originated from the fact that such a large percentage of drugs are delivered orally, and thinking about the gut as being that key barrier and characterizing how well does a novel drug get absorbed or metabolized in the gut, in addition to the fact that there is such an important market related to inflammation in the gut. Really, all of those factors helped catalyze the rationale to build and to continue to develop better gut-based models.
That's great. And we'll dive into a little bit more on that, but that's wonderful. How has your leadership approach evolved as Altus has matured now from concept to more of a commercial platform?
Yeah, I think your question hits the mark squarely in terms of building an entity that is commercially focused, is really making sure that a team keeps things in mind of we work to serve customers. Altus is really in the CRO type of space. We provide services, we provide customers, and the perspective for us is definitely not just building the best tools possible, but building tools that help address customer pain points and where there are really needs for these types of advanced model systems in preclinical drug development. And from a leadership perspective, I really think of that as making sure that everyone on our team is always thinking from the perspective of what are those needs of the customer that we should be thinking about, and how can we continue to improve our products or expand our offerings in ways that are addressing real gaps in the market. Um, from that perspective, it's instilled a notion of we focus on simplicity in terms of what we do and building things that are intentionally simple models. We don't necessarily try to build the most complex models possible to address those pain points. And thinking about how do you build things from the ground up is kind of the approach that we take. And being able to iterate on that. And I think that's an aspect from a team perspective that we try to really focus on and keep top of mind as we try to address the pain points before anything else.
That's great, Ben. So simplicity for sure. What is, I guess, building a science-driven company taught you about hiring and culture?
Yeah, building a science-based company is there's a balance of everyone who comes from any type of scientific background tends to share a couple of common threads. One of those that's probably top of the stack is scientific curiosity or just curiosity in general. I think that that is the spark that is really necessary for a successful life science company of any flavor is being able to have that room to continue to push boundaries, but doing it for a specific reason as you're building a company. The company has to balance that scientific curiosity with focus. I think that that is part of the primary responsibility of leadership is to make sure that you give enough room to keep that spark going. It's really the fun part of working in our industry is continuing to innovate, but doing it in a way that's constructive in terms of making sure that it's for the specific goals of a company to push that forward, and in many cases as a commercial success.
Excellent, excellent. That's well said. Balancing that creativity with focus, as you say. Let's talk a little bit about more about the company technology. We've touched on this, but I guess if you could, the company's product replica, we'll explore that a bit. But just generally, what limitations exist in traditional intestinal models today?
Yeah. It's interesting because in vitro intestinal models have existed for decades. So while there's quite a bit happening in advanced model systems right now, be that anything in new approach methodologies, be it organ on a chip or organoids, there's a lot happening. But the gut is interesting because different cell types have been used for decades. They've been recognized by the FDA as part of submitting an IND package, to be able to run some of these studies, either with Keiko2 cells or MDCK cells. They're different flavors of what's used out there. But what's been interesting is some of those different cell lines that exist still don't faithfully recapitulate human biology. The limitations that exist there, just using an example of Keiko2 cells, are actually a tumor-derived model. And they have some deficiencies in terms of having metabolic enzymes present in those cells or transporters that would be used to transport drugs across the epithelial barrier. Those aren't necessarily expressed at human physiological levels. And they certainly work as a tool to be able to look at whether or not a drug might be permeable across the membrane, but they're not necessarily accurate in predicting all aspects of a wide class of different types of drugs. And so they aren't true physiological representations of human biology. And I think that's where we've really seen, others have certainly seen that there are opportunities for improved models that could better represent the way that human physiology really works. And that's how we think about things, again, from that simplistic standpoint. And I think the other aspect is that there are key components that interact with that epithelial barrier, be it immune cells or other interactions that really create the full representation of human biology, and integrating that into systems to answer key questions in drug development are where there's still been a gap, even though there have been tools like what I just described that have existed for a while.
That's great. That's great. And so replica itself improves physiological relevance, it sounds like.
The idea is intentionally simple. The idea is that we take full-length human intestinal tissue. So we take that intestinal tissue. In the intestinal crypts, there are primary stem cells. So they're specific to the intestine. We basically have a procedure where we shake out those stem cells, quite literally, and pull those stem cells out. And that's our core starting material to create a system. And what our system looks like is we take those stem cells, we put them on a scaffold that's pretty straightforward and familiar to anyone who's done cell biology in the lab. And we can differentiate those stem cells into all the different cell types that make up the epithelial barrier. So these cells differentiate into the relative cell populations that make up the lining of the gut and their relative populations that you would expect to find natively in the body. And that allows us to create a system that assembles and has a barrier that we can measure, we can disrupt. And so it's very amenable to a number of different measurements and downstream measurements to specifically ask questions, be that mechanistic questions for drug development or really understanding sort of the drug metabolism, pharmacokinetics aspects of drug development. It's all of those are applicable, and it's all because we're starting with that human material initially.
Absolutely. No, that's great. What types of pharma or biotech partners, I guess, benefit the most from the RepliGut platform?
Yeah, it's a pretty broad audience of who can use a tool like this. I think there are a couple of different categories of who primary users are for the RepliGut tool. Um, Historically, a lot of the work that we've done has been with customers that are working in the inflammatory space. So, inflammatory bowel disease, for example. Certainly, a lot of different products that have been developed in that space. Historically, you look at drugs like Humira have been some of the best-selling drugs that have existed. But when you think about drugs like anti-TNFs or the JAK inhibitors, while certainly fantastic drugs and have been efficacious for a number of patients, those drugs are effectively like sledgehammers. They just bluntly knock down inflammation. And what's been unfortunate about those types of tools is that a lot of patients, they either don't work for all patients or patients become refractory to those types of drugs. And so we've done work with many companies that are developing the next wave of therapeutics for inflammatory bowel disease. There's quite a lot of work that has gone into things related to precision medicines in IBD, or some of the newer waves, drugs like the TL1As, and whatnot. So quite a bit of continued development there. And so that's broadly one category. But I think the other aspect that we've really had appreciation for, and probably where we're focused more than anywhere else at Altis right now, is simply looking at the absorption and metabolism of orally delivered drugs. So not specific to one therapeutic area, but looking at oral delivery, thinking about this from the mindset of bioavailability of drugs. There are multiple components that go into how well is an oral therapeutic going to be delivered to its target of interest. I think historically, most of the work has happened in the liver. And part of that has been because the gut, while it also has a significant contribution, the models haven't been very accurate in predicting that. So that standpoint has really been what we've focused on. So we break it down in terms of thinking of the gut's contribution of what fraction of that drug gets absorbed, what fraction of it gets passed to the liver, and then ultimately the bioavailability is that contribution of both the gut and the liver to a drug entering circulation.
Very good. Thank you, Ben. Let's talk a little bit about the economics. How does the improving gut modeling change the economics of drug development?
Yeah, I mean this is the part where things really get interesting. Is there are multiple different aspects. Certainly, being able to better predict human biology before you take something into the clinic is where the major savings can occur. Certainly, and we've had several clients who have taken compounds into the clinic, and they may get some sort of different um, pharmacokinetic profile than what they originally expected, despite the fact that they measured that in multiple species before they took it into the clinic. But you're talking, of course, the order of millions to run those types of clinical trials. Um, and that's really where the biggest impact can be. I think the other aspect is savings in time and being able to get answers quickly because we're using a cell-based model, and rather than relying on animal models to better understand aspects related to absorption, metabolism, or inflammation, when you're answering questions on the order of weeks rather than months, for animal models, that of course is going to save money, but it will also save time. Using a cell-based model is going to be cheaper. But I think I would really break it down in terms of couple of different stages of preclinical development. Lead optimization is really the aspect that I would say is the sweet spot for most of the work that we do, of being able to really help a company define that they have a drug that is ready to be nominated as a development candidate, for example, and that is a sweet spot. But I think more broadly speaking, sort of the hit to lead aspect is where we see what we focused on at Altis is being able to do things that are relatively high throughput. So we design these assays to be done in multi-well formats. So you can test several different compounds. In some cases, we have users that are going into the order of testing thousands of potential compounds at one time, and being able to triage that is really a major savings in time, which obviously economically leads to savings in cost if you can triage which compounds to prioritize over others.
Absolutely, which to target. That's great. Let's go into the scaling kind of business strategy aspect of the business for a few moments. What have generally been the hardest scaling challenges, I guess, for the company so far?
Yeah, easily. The answer to that is reproducibility. So it has been, if there's been one defining aspect of our company, it's been to focus on reproducibility. We work with primary stem cells. Intrinsically, they are prone to variability. That's part of it. We're not working with a cell line in the traditional format. So the ability to put a quality control procedure in place where we can ensure that we're going to be able to manufacture cells, which at our core is part of what we do, that we can do that in a reliable, reproducible manner is really what allows us to manufacture cells at scale and have allowed us not just to work as a service provider, which is a big part of what we do. We have a team of exceptionally talented scientists that can design studies for users, and often in biotech, that's what customers want, is they want to outsource the work and have people do that for them. But you almost have an equal proportion that would prefer to be able to use the product in their own labs. And so the focus on reproducibility has allowed us not just to run our own services, but effectively take that same RepliGut product and ship it anywhere in the world. And from that standpoint, the results that we would generate with a study in our hands, it works the same way in other users' hands when we ship that out. And that's certainly been game changing for us to be able to have that dual business model of being able to do both. But it focused on quality and reproducibility to be able to enable that.
Those were core for the to enable the dual business model. That's great. How do you prioritize, I guess, platform expansion versus, importantly, revenue generation
Yeah.
for the company? Those things are, I guess, sometimes at odds, but
Yeah, no, it's a daily question, and it goes back to what we talked about, that balance of innovation and focus as well. I think that the aspect of what the way that we do it is as we listen to needs before anything else is where are there gaps in the market that we see as really needing new tools to exist. And when we're listening to clients that tell us that their key questions when we hear something on repeat again and again, then it's really clear that we need to develop something new for that. There are different areas where those types of things pop up. We think about that all the time. We think about that, to think of a topical area, things like GLP-1s are not lost on us in terms of some of the things we've described. Bioavailability can be limited for some of those new oral products that are coming out. And so those are things that we think about internally of what do we need to demonstrate or what could we potentially offer that would actually have utility in the market. And when we see opportunities like that, that's when we decide that it makes sense to launch new products. And we've done that pretty consistently over the past 18 to 24 months, is continuing to come to market with new products that we think really offer additional value to users.
that's great. That's great. Thank you for that. I'm gonna turn and ask you to put on your investor hat for just a moment here. So a couple of questions. What advice would you give founders building infrastructure technologies in biotech, and in conjunction with that, of course, what matters most to investors evaluating these platform companies?
Yeah, I think that for founders, I think that I'm going to go back to this idea of simplicity. I mean, this is one thing that we can learn from our colleagues in the tech sector of minimal viable product is real in spaces like this of trying to identify what is the core unit that something can look like to really be able to address needs, and defining that and making sure that you get that as strong as possible before trying to tinker and add complexity to a system. I think one of the things that's really interesting about this new approach methodology space is there are a lot of different aspects of it, be it organ on a chip, organoid, advanced in vitro systems like ours. They're all fit for purpose. There's utility to all of those things, but when you're building, really understanding where is that gap that you're trying to address. I think on the flip side of that, for investors, it's demonstration of utility is really the core aspect to evaluate. Is what is the competition really look like in this space? I think just to give an example from Altis, and in our early days, a lot of thought was competition relative to other players. I don't believe that to be true at all. I really think that the space we're in right now is a rising tide raises all boats. Some of that we've learned of. We've done collaborations with other model system companies. We have companies that are using our cells on different platforms. It's an aspect of how can we do drug development better in general. And that's really, I think, something that's important for investors to think about as they're evaluating opportunities right now. We talked about some of the existing cell types that are used in the market right now. What are those limitations? For the gut, we've laid that out, but there are limitations for all sorts of different systems. And human-based models, I strongly believe, have a real place in the market. It's just trying to understand what questions can you answer with this that there's a need for, and how will that make drug development more effective? And I think that's what I'd love to see investors really focusing on in this space.
That's great. Just to dovetail on that, what as we think about the future, what collaborations excite you the most at this point? And I don't know, could you see predictive human gut models, reduce clinical trial failures, and maybe even, I don't know, replace animal models one day?
Yeah, I think that the notion of replacing animal models is it's a great idea. I really think that we're quite a way away from that. I do believe that we are in a great place to reduce the reliance on animal models. We can be much smarter about which drugs, which doses do we actually take into animals so that we can do fewer studies in animals. I think that we are on the cusp of that, if not there already, to be able to do that. Um, the thing that I'm most excited about from a collaboration standpoint is really the intersection of not just some of the advanced tools that exist, but we're generating higher quality data with these models. And so, how can we use this data as input that can help us make better decisions as we go into the clinic? And so, collaborations that really make the intersection of advanced models intersect with computational methods, I think are really interesting. So, in vitro meets in silico is a really interesting pattern. And I think that there are opportunities for that. There are a lot of interesting companies that are emerging in the in silico toxicity prediction space. But I'm thinking of things where if you're generating human-based data, there's value to that that you could put into a model, a computational model now that could effectively allow you to run a human-based predictive algorithm before you take anything into the clinic. And so I think all of this could eventually become a way to better design clinical trials. That's really what I would hope that this all leads into. And again, that's going to go back into your excellent question before. Economically, that makes all the sense if we can run better trials that way.
Absolutely, potentially millions and tens of millions of dollars saved, I'm sure.
Yeah.
Just is there any other advice you would offer, Ben, to founders building the next generation of life sciences biotech infrastructure? Any other general guidance?
Um, it's a great question. I think that my biggest advice is spend time talking to drug developers and really understanding where where are the gaps, where are the biggest challenges that we are facing right now. Talk to drug developers at all stages, at the earliest stages from target discovery all the way through to preparing IND packages, as well as those that are reviewing some of the clinical data. I think that those aspects are imperative to understanding how to build the best companies possible and not just get focused on building a really exciting scientific tool. I think that you have to start with the end user in mind as really the place to build. And I would say doing that in a really disciplined manner. I think that capital efficiency is probably more important than it ever has been. And so, understanding what is the place to try to commercialize products first, is important to be able to demonstrate that you can generate revenue from these types of tools before just trying to build every possible iteration of a scientific idea. So I guess my takeaway from that is less is more, but having it as a really clear idea of who you're trying to solve a problem for.
Absolutely, and listen to your customer base as well.
Exactly.
That's great. Finally, how can listeners connect with you and learn more about your work at Alta Systems?
Yeah, so several different ways. Certainly, altisbiosystems.com has a full description of all of our product offerings, our core RepliGut, as well as the iterations that we've made on top of that. And always happy to connect via LinkedIn as well. Please feel free to reach out. We have a team that's truly committed to working with customers and even just talking about ideas of the best tools out there that could potentially help anyone doing drug development.
Fantastic, Ben. Thank you. Well, that wraps up this episode of Executive Insights for Life Sciences Innovators. I appreciate you tuning in and hope you found today's conversation valuable. If you enjoyed the discussion, please be sure to leave us a review and share it with your network. Please also mark your calendars for March 9th at 1 o'clock Eastern with Chris Hoyt of Keith RX.